Thank you for inviting me here to speak in this wonderful place. And today we're going to change tact and start to think about mice and men. So today what I want to do is talk about the effect of early life nutrition on the epigenome and its implications for later health. So traditionally it's been widely accepted that our genes determine our phenotype and susceptibility to disease. But it's becoming increasingly clear that the environment, particularly early life environment, can also influence disease risk. And one of the first suggestions that early life environment might be important for future disease risk came from the work of David Barker, who showed that low birth weight was associated with an increased risk of both heart disease and metabolic syndrome in mid to late life. Since then, there have been numerous epidemiological and experimental studies, and these have confirmed these observations and also gone on to show that an adverse early life environment induced by placental insufficiency, maternal undernutrition, overnutrition, maternal obesity can all perturb the maternal fetal environment and lead to long-term metabolic changes within the offspring, often resulting in an increased risk of chronic disease. The mechanism by which early life environment has been suggested to induce these long-term metabolic changes within the offspring is through the altered epigenetic regulation of genes. And these, as we've heard, are processes that can induce long-term stable changes in the expression of genes without a change in the gene sequence. The three main epigenetic processes are DNA methylation, histone modification, and non-coding RNAs. And to date, most of the focus has been on DNA methylation in this field. So in mammals, when DNA is methylated, it is most commonly the cytosine base that is methylated. The majority of these methylated cytosines are found as part of the dinucleotide CG. And in general, low levels of methylation are associated with the transcriptional activity of a gene and high levels with the transcriptional repression. What we also know is that the methylation status of an individual gene is largely established during development or early life. We know that upon fertilization, the methylation marks on the paternal and maternal DNAs are largely erased. Then, as development differentiation proceeds, there is this gradual increase in methylation, and genes that are not required in a particular cell type are methylated and switched off. And this essentially creates tissue-specific patterns of gene methylation and expression. And it is this that essentially creates our distinct cell types. But once this period of development, plasticity is over, it is thought that these methylation marks are relatively stable and maintained throughout our life. But given that one carbon metabolism, which provides the methyl groups for virtually all these DNA methylation reactions, is largely dependent upon dietary methyl donors and cofactors, it has been suggested that maybe perturbations in maternal diet, nutrient availability during this key early phase of life might perturb the establishment of these methylation marks, lead to a resetting of these marks and long-term changes in the expression and metabolism within the offspring. So we wanted to test this, particularly to see whether perturbations in maternal nutrition might alter the methylation of key metabolic genes within the offspring. So we fed pregnant rats a modest reduction in protein just during pregnancy, and we found this altered the methylation of a number of metabolic genes, including the PPAR alpha gene, which plays a key role in lipid metabolism. So what we found was there was a decrease in methylation across the promoter region of this gene in the liver of the protein-restricted offspring compared to the controls. This decrease in methylation was accompanied by an increase in the expression of this gene and an increase in levels of beta-oxidation, the metabolic process that this gene controls. We found this change in methylation occurred as early as embryonic day 18 and persisted right the way through the life course. But it's not just maternal undernutrition that can induce these epigenetic and phenotypic changes. We've also seen that maternal overnutrition can also induce such effects. So here we fed pregnant rats a diet either high in saturated fats or in fish oil, again just during pregnancy. And we found this altered fatty acid composition within the liver of the adult offspring. But specifically, we saw a decrease in 22, 6N -3 and 24N -6 levels in the liver of the offspring from the dams fed the diet high in saturated fats or in fish oil. As the FADS2 gene encodes the rate-limiting enzyme in the synthesis of these fatty acids, we then looked at its expression and methylation within the offspring. And we found there was a persistent decrease in the expression of FADS2 in the liver of the offspring from the dams fed the diet high in saturated fats or in fish oil. And this decrease in expression of FADS2 was accompanied by an increase in the methylation of a number of CpGs within the promoter region of this gene, including the CpG located at -394, with methylation at this site being associated with over 56% of the variance in levels of FADS2 mRNA expression in the individual offspring. But we and others have also shown that variations in early postnatal nutrition, as well as variations in paternal diet, can also induce epigenetic and phenotypic changes within the offspring. Such changes to the methylation of genes that often play key roles in metabolism and appetite control suggest then these changes may well underpin the long-term changes that we see in expression and metabolism in the offspring and lead to this altered disease risk. Interestingly, there's also been studies which have shown that maternal diet can induce metabolic changes that persist through to the F2 and in some cases to the F3 generation. So in this study here by Tracy Bale, they fed a high-fat diet just during pregnancy. And they found this induced extensive reprogramming in the F1 offspring. The majority of these effects dissipated in F2, apart from the effect on body length and body weight, and this was maintained through to F3. But interestingly, it was only maintained in the female offspring through the paternal line, suggesting that there is some capacity for transgenerational transmission, but maybe this is dependent on exposure and the trait. But what then about us? Does the same phenomena occur in man? Well, a number of groups have been looking at subjects from the Dutch hunger winter, because back in the Netherlands in 1944 there was a famine when energy intakes dropped from 1,800 kilocalories per day to between 400 and 800. And what subsequent studies have shown is individuals born to women exposed to famine during pregnancy have an increased risk of developing heart disease, diabetes and obesity in later life. They've also interestingly seen increases in adiposity in the grandchildren as well. But what they've also seen is alterations in the methylation of a number of genes in those individuals who were in utero exposed. Specifically, they've seen an increase in the methylation of interleukin 10, leptin, and a decrease in the methylation of IGF-2. And interestingly, these measurements have been made 60 years after the famine, suggesting again that variations in maternal diet can induce these very persistent epigenetic changes within the offspring. So if the same phenomena does occur in us, and that early life environment, nutrition can alter these methylation marks on our genes, leading to or being associated with long-term changes in metabolism, then it might be possible to detect such changes in early life and use these to predict later metabolic capacity and disease risk. The major issue with doing this in humans is there is limited tissue that we can collect. So tissues that we can readily access would be cord blood, placenta, buccal and blood. And there's a real question mark whether the methylation in these cell types adequately reflects the methylation in more metabolically relevant cells. But maybe if the environmental constraint occurred early enough in development, then all three germ layers might be altered. So we decided to look to see if we could find methylation differences at birth in cord that were associated with later adiposity. So we carried out a genome-wide screen, and we found there were 93 differentially methylated regions at birth in cord associated with percent fat mass of the children aged 6. And one of the marks that replicated across 2 independent cohorts was a DMR which was located within the SLC6A4 gene, which encodes for the serotonin transporter. And here we found that lower methylation of SLC at birth was associated with a higher percent and total fat mass at 6 years of age. We saw this association in cord and also cord blood, suggesting that maybe there is an imprint on the epigenome that we can detect across tissue types, and maybe we could use these marks then to identify individuals at increased risk of adiposity and obesity. But this ability of early life environment, nutrition to alter these methylation marks has been suggested to be part of a normal adaptive process, a process by which an organism can respond to the environment, adjust its developmental path to produce a phenotype that confers a survival or fitness advantage. So, for instance, poor maternal diet might signal to the fetus that nutrients are scarce, so the fetus will then adapt its metabolism, reduce energy demand, increase its propensity to store fat. If then it finds itself in an environment where nutrients are indeed scarce, its metabolism will be matched, it will be of low disease risk. But if it finds itself now in an environment where nutrients are actually abundant, its metabolism will be mismatched. It'll have this increased propensity to store fat, be an increased risk of metabolic disease. And this mismatch between the pre and the postnatal environment has been suggested to account for some of the rapid rises we've seen in rates of obesity and diabetes in the developing world as populations move from rural to urban areas. But what then about the Western world? Because here we are being exposed, now at least, to a sustained nutritional challenge, to a high energy, high fat diet. Can we adapt, or are such diets outside our adaptive capacity and will lead to a cycle of obesity? So this will perhaps lead to maternal obesity, then the women will give rise to children with an increase in adiposity, which then tracks into adulthood. So we wanted to look at the effect of the sustained nutritional challenge. So we took mice that were normally fed a chow diet, which has a 2.7% fat content, and we placed them on diets with an increasing level of fat, ranging from 5% to 21%. The mice were fed these diets during the F0 pregnancy, and then the offspring were maintained on these diets. And when we looked at weight gain, we found there was a significant increase in weight gain in the mice fed the 21% fat diet in the F1 generation, but no difference in weight gain in F2 or F3, suggesting that there is some adaptation or adjustment to this high fat diet. But when we looked at weight gain in the older mice, we now saw there was a significant increase in weight gain in the 15% and 21% dietary groups in F1, F2 and F3, suggesting whatever adjustment capacity there was, this was now lost in old age. So we've started to look at some of the molecular changes in these mice, and today I just want to tell you briefly about some of the changes we've seen in the expression of genes within the liver of these mice. So first of all, we looked at the effect of diet, and we looked first in the F1 generation and on day 90, and we found there were 653 genes that were differentially expressed between the mice fed the 21% fat diet compared to those fed the 5% diet. But when we looked in the F3 generation now, we saw that there were far fewer genes changed. Now there were only 23 genes differentially expressed between the two dietary groups, suggesting that over time these two dietary groups have converged. So to understand this convergence, we then looked at expression of the genes between F3 and F1 in the two dietary groups. And we found when we looked in the 5% dietary group, there were 511 genes differentially expressed between the F3 generation and the F1. But when we looked in the 21% dietary group, again we saw only 17 genes differentially expressed between F1 and F3, suggesting that the changes induced by the 21% diet during F1 are largely then being maintained through to F3. So this then suggests that over time, the 5% dietary group is becoming more similar to the 21% dietary group. But what then about aging? So we first looked at aging in the F1 mice, fed the 5% fat diet, and we compared genes expressed on day 456 to day 90, and we found there were 892 genes differentially expressed. The top pathways enriched amongst these genes were oxidative stress, acute phase signaling, and mitochondrial dysfunction, pathways that were very similar to those seen in liver aging in our chow-fed mice, and very similar to the pathways seen in a recent meta-analysis of liver aging. But when we looked at aging in the mice fed the 21% fat diet, we now saw that there were only 99 genes differentially expressed, and a similar number of genes changed in F3. So we wondered then whether this lower number of gene changes that we see in the 21% group and in the F3 mice was due to the fact that the pathways affected by aging, diet and generation were in fact very similar. So we compared the genes changed by diet and aging. So what we did was to compare the genes changed by diet and also aging in this 5% group here. And what you see is of the 653 genes changed by diet, 579 of these are also changed by aging, and the vast majority of these are changed in the same way. So given that generation also affects a similar set of pathways, we then looked to see whether there were genes commonly changed by aging, diet and generation as well. And we found there were 400 genes commonly changed by age, diet and generation. All 400 genes were changed in the same direction, although the magnitude was different between these drivers. So this suggests then that a high-fat diet, feeding a 21% diet, induces changes in the liver that resemble that of an aging mouse. But perhaps more interestingly, that even feeding a moderate increment in fat also induces those same changes, a bite over 3 generations. So in summary then, there is growing evidence that maternal nutrition can have a significant impact on metabolism and health of the offspring. And there are cases where this can be transmitted through to the F2 and perhaps the F3. There is also growing evidence that early life environment can alter these epigenetic marks. And perhaps we can detect such marks and changes and use these to identify individuals of increased risk of disease. But it should be said, at present what we have is we have associations. We have an association between a change in methylation and a change in phenotype. We do not know whether this is causal or whether it is simply a mark. But what we have also seen is that now we are being faced with a sustained nutritional challenge. And from our mice work, it would seem that even being exposed to a moderate increase in fat over a prolonged period of time can have a deleterious effect. So what is clear then is that we're beginning to enter this cycle of obesity, and it is now becoming very important that we understand how these traits are passed from one generation to the next, whether these epigenetic processes are involved, how important they are. But perhaps by being able to detect such changes in methylation, whether they are causal or not, we can begin to identify the individuals at risk. And perhaps we can even use these changes to also identify the agents or environmental factors that can potentially change risk as well. So I'd just like to finish by acknowledging the people that did this work, but particularly Paula, Emma and Hazel. Thank you.
Vincent. Here, sorry. A very simple question. So you have the influence of the environment on some traits, correlated traits, aging, fat amount, etc. But now, what happens if you allow these animals to grow without the high fat diet, or do you still see the phenotypes, or is it completely reversible?
The most of the traits disappear. Yeah. So
can we conclude that there is no transgenerational effects in that respect? So basically, the environment does matter to the individual that receives these insults, but it doesn't leave long-lasting consequences for the progeny?
Yeah, I think so. In the study that we did at the end, that was a 21% fat diet, and with that diet we didn't see any true transgenerational effects. In other studies, people have used a much higher level of fat, and there you do see some effects through to the F3. So I think it depends on the severity of the challenge.
I want to ask a question in the same direction. If you do this experiment with the 21% and you don't see any effect, but then you expose them again to 21% or even 5%, is there a difference in terms of the response to the stress? In other words, is what you're doing, although it doesn't have a phenotypic effect, transgenerational is nevertheless priming the mouse to respond to such a stress in a different way?
Okay. Yeah, we would love to do that, but we haven't done that yet. But I mean, that would be really good to do. Yeah.
Jean-François, back.
I have two questions. The first one is, have you looked at generations further than F3? How many generations will you see the effect? And the second question, you essentially look at methylation. Have you looked at other epigenetic marks, such as other mechanisms, such as non-coding RNA, or just focused on methylation changes?
We've only looked at F3 to date. In terms of other mechanisms, what we do see, if you look at PPAR alpha, is you do see a change in histone modifications before you see a change in methylation. So the methylation change seems to be consolidating the change in expression that is brought about by a histone change. But we haven't looked at the involvement of long non-coding RNAs.
Moshe Yaniv. On your back.
When you give a pregnant mice a high-fat diet, is there an effect of the placenta? Does it filter? Does it kind of a barrier for metabolites?
Yeah, we are looking at transfer across the placenta of the high fat, and we're just doing that work now. So because there's been quite a lot of controversy about how much actually gets transported across, so we're doing that work now.
I'm very naive on this topic. From your experiment, can you detect some sensitivity of the origin of the strand of viruses that you are using? I mean, transposing to human. Are Caucasian or Chinese? Do they have the same sensitivity to environmental differences or stresses? Do you understand what I mean?
No, could you?
No. Do you think that Chinese or Caucasian population could display exactly the same response to stresses, or do you expect that there will be differences due to the past history of both civilizations?
So we do have a cohort which is based in Singapore, and that has Malays, Chinese and Indians in that cohort. And we also have cohorts in the UK. And so what we do know is that the methylation changes we see, say, with SLC6A4 are seen across all those populations. So we're looking now to see whether there is a common intrauterine environmental factor that might explain the differential methylation of that gene that might be common to all those populations. But I think it is likely that there will be differences. There are clearly big genotype differences. But in fact, if you look at methylation across all those populations, what you see is a very mixed pattern of methylation. So it doesn't cluster depending on the genotype. So the methylome, if you like, goes across and is a very mixed kind of picture. Yeah.
Thank you.
If there is no further questions, thank you.